much for inviting me to share our work with you today and for all the support that the CFRI has given us over the years in generating the data and ideas that I'm going to be telling you about. I was asked to talk about phage therapy, and so I'm going to take sort of a broad view of that and actually tell you two stories. One story about targeting a bacteriophage that we think causes disease in cystic fibrosis, and then a second set of stories about generating phages to target bacteria that we also think cause, well, we know, of course, cause disease in individuals with CF. And so I'm calling this a tale of two phages. Okay, so as you folks know, I'm sure better than anyone that there's been remarkable progress in CF clinical care, but there are still many gaps that we need to address. In particular, chronic lung infections remain a problem in individuals with CF, both in terms of folks who were diagnosed before HEMT therapy, but also in individuals who are receiving this, who nonetheless continue to have chronic infections, quite often with Pseudomonas aeruginosa, but also Staph aureus and other organisms as well, as you can see here in this recent study. In particular, one bug that has remained a problem despite HEMT is Pseudomonas aeruginosa. And Pseudomonas is going to be very well known, I'm sure, to both individuals with CF and people who care about them. But this is a highly problematic organism for several reasons. The first is that it causes biofilms, which again is a concept that I don't need to explain to this audience. And this leads to impaired mucociliary clearance, where it's difficult to mobilize these biofilms and to clear the bacteria that causes them. Pseudomonas is also problematic because of the lack of effective antimicrobial therapies. Pseudomonas is a bad actor for several reasons. The first is that it causes biofilms. This leads to impaired mucociliary clearance. And then over time, the antibiotics that we use to treat Pseudomonas select for really nasty strains of Pseudomonas that are resistant to many of those antibiotics, and in some cases to all antibiotics. And so in my lab, we've been asking two questions. The first is, what makes Pseudomonas so problematic in individuals with cystic fibrosis? And then what can we do about stopping antimicrobial resistant strains of Pseudomonas? And I'm going to offer that bacteriophages are potentially the answer to, or an answer to, both of these questions. And so what are bacteriophages? Well, there's a picture of them right here. And so phages are, well, let me see, as you guys are seeing the regular view. Okay. So phages, it's okay. All right. So phages are viruses, just like you and I have viruses or exposed to viruses out there in the world. Bacteria are too. The only difference is that bacteriophages parasitize bacteria rather than, their human hosts. Phages are highly strain specific. So a phage that infects Pseudomonas isn't going to infect Staphylococcus. And for that matter, a strain of a phage that infects one type of Pseudomonas isn't going to infect another type. And we'll come back to why that's so important. And then phages are abundant at sites of bacterial colonization. So you and I sitting here, we have more phages in our body than either bacteria or eukaryotic cells. So in your gut, in your skin, in your mouth, you have phages all over the place right now as we're all sitting here. So kind of remarkable. And phages come in two flavors. The first type of phage, if folks in the audience are familiar with phages, is probably the type that you've heard about. That is to say, phages that infect bacteria, just like, say, influenza might infect a eukaryotic cell, they use the cellular machinery to make more phages, and then they break out and go on to infect the next round of bacteria. And these are called lytic phages. And I'm going to offer in the second part of my talk that these are potentially an exciting solution for antimicrobial resistant bacterial infections in the CF lung. But there's a second type of phage, and these are called lysogenic phages. And unlike the lytic phages that infect bacteria and kill them, lysogenic phages are more complicated. They can live in bacterial genomes, and they transmit things like toxins and other genes that cause problems. And then they lyse their hosts opportunistically. And so I'm going to tell you folks about two stories, and then in parallel, two types of phage therapy, if you will, that we can use in each of these two stories. The first story is about a lysogenic phage. Again, these phages that live in their bacterial hosts and partner with them to cause disease. And I'm going to tell you about one that causes poor outcomes in cystic fibrosis. And then I'm going to tell you a second story about lytic bacteriophages that we can use to protect individuals with CF from bacterial infections. Okay, so the first story about lysogenic phages has three parts to it. The first is the idea that bacteria and phages can partner together to build biofilms. Then I'm going to tell you about how this partnership protects Pseudomonas from antibiotics. And then I'm going to tell you about a vaccine that we've developed targeting this phage, so a different kind of phage therapy, if you will, that we're optimistic may one day be able to prevent Pseudomonas infections. So let's talk about that part. What got us down this road was the question that I'm sure many of you have already asked, which is, how does Pseudomonas do this? How does it make biofilms? And why are these so problematic in cystic fibrosis? And so biofilms, for those of you who aren't familiar with them, are big slimy matrices of bacteria, but then also the polysaccharides that they make. And so in the same way that maybe a termite mound is not just the termites, or an anthill is not just the ants, it's the actual substrate that they live in. And you can sort of consider, the dirt and the termites here as one organism. And similarly, biofilms aren't just the bacteria, they're also the bacteria and the polymers, the substrate, the matrix that they produce. Yes?
I was just curious, the image in the top left, the sludgy stuff, what is that?
So this image right there is a sample of CF sputum from, optimistically, another era.
Okay.
And so this was, I'm 53 years old. I started this a long time ago.
Yeah.
And I've had this slide in my deck. But I'll show you some other pictures too.
Okay. It looks very interesting.
It's very viscous, right. And you'll notice that it's also rust colored, right?
Yes.
So you've got a bunch of blood in there in all likelihood.
Oh, yeah.
And, I actually, on a personal level, I remember being 7 and having a friend in my class with a younger brother who had CF, and being at their house when they were doing, all of this pounding, on the back to mobilize samples like this. And, after 20 minutes of this or whatever, it was like waiting for Elijah, like this thing would finally come up. And yes, so sputum from another era. But the point here is that I think of this as sputum, or at least I used to think of this as sputum, but actually it's a microbial biofilm. Like it's not just saliva and mucus, it's an actual organism that has set up shop in the CF airway. Okay, and so the question we started asking in my lab is how does Pseudomonas do this? How does it make these biofilms? And so a postdoc who we were working with named Pat Secor in a lab down the hall did this really interesting experiment where he took some hyaluronic acid, which is a polymer that's present in the lung, and he added it to a culture of Pseudomonas. And he saw that it made these funny structures right there. And that struck him as being really odd. So he added some hyaluronidase, and the structures basically dissolved. And we thought this was really cool because it suggested that Pseudomonas can actually grab polymers that are out there. Polymers are long strings of carbohydrate. DNA is a polymer. Mucus is a polymer. And Pseudomonas can grab these things and build them into little structures that are almost like houses, if you will. And we thought this was really cool. Nobody had seen this before, to our knowledge. And then we learned that you didn't actually need the Pseudomonas. You could actually just take the supernatant, which is the top layer of a bacterial culture, and you can add that to a solution of polymers, and you get this thin adherent film that you can see here. And so what this is actually a glass cover slip, so a little piece of glass that Pat added some supernatant from Pseudomonas and some hyaluronic acid. And then he took a little nail, basically, and ran it down that glass cover slip, and you can see that it made a trail, right? And so this is, looking at that in another way with a different kind of microscope, But the point is that Pseudomonas makes something that floats around and can grab polymers and make adherent films. And I'm going to argue in a minute that it does this in our lungs. And after doing a bunch of investigation that took an embarrassing number of years, we figured out that you don't need the Pseudomonas, you don't need the supernatant. It's actually this guy here, which looks like a little thread, or maybe like, I don't know if anybody knows Charlie and the Chocolate Factory, like the Firminius canids or whatever. But this little thread is actually a virus. And I'm going to tell you in a moment that Pseudomonas uses this virus to build biofilms. And I'm going to talk about how we can defeat that. Again, one form of phage therapy, if you will. Now, these phages aren't like the lytic phages that we can use in lytic phage therapy for bacteria that blow up bacteria. Instead, what these guys do is they bud off of the surface of the Pseudomonas, and Pseudomonas regulates their production. And I'm going to make the argument that Pseudomonas has basically domesticated a virus that it uses to build biofilms. And so what we found is if we took this phage, this virus that Pseudomonas makes, and we mix it with any type of polymer that we've identified in the body or otherwise, it will spontaneously self-assemble into structures that are basically, again, these adherent films. So this is a polymer called collagen that might be in some of the skincare products you buy. This is DNA, which is another type of polymer. This is serum, so the stuff in your blood that isn't cells. And it does the same thing with sputum, with urine, with other polymeric solutions. So the question is how does this work? And so one clue that we got was we took a solution of alginate, which is a bacterial polymer that's present in biofilms. And we took a drop of this phage and added it right here. And as quickly as Pat could take the picture, it essentially made these structures that took over this bacterial or this slide. And so I'm a real science fiction geek. Has anybody read the book Cat's Cradle? Long time ago. Okay, so there's a molecule in Cat's Cradle called Ice-9. And Ice-9 is a crystal, and crystals have this very favorable state of entropy, if you will. And so a little like a rock being at the top of a hill versus the bottom of the hill, all rocks will eventually fall to the bottom of the hill. They want a favorable state of entropy. And so crystals, like ice and water, have a funny way of organizing their environment around them into more crystals because it's entropically favorable. And so in Ice-9, the whole world has basically turned into a crystal. And when I saw this image here, that's kind of what I remembered, this book. And sometimes science fiction really informs reality. And so we wondered, are these structures that we're seeing, and I'll show you in a moment that these things are also in the CF lung and in CF sputum, we wondered, are they crystal? Are they crystalline? And so one property of crystals is that they're birefringent, a little like your polarized glasses. They allow light through in one direction, but not another. And so you can look for birefringence to see if something is crystalline. And so we looked, and polymers like alginate, again, that Pseudomonas makes, are not crystalline. This phage we identified that Pseudomonas makes are not crystalline. But if you mix these two things, all of a sudden you get something that is birefringent. And you can make these really cool phase change diagrams where you have the concentration of polymer and the concentration of phage, and you essentially get a phase change diagram. And some of you guys may have PTSD from high school, and you remember like water turning into ice, has a very similar kind of curve like this. There's a point where, essentially a liquid turns into a solid, and that's what happens with crystalline materials. And so we thought this was cool because it told us that PF-phage assembles structures like this into a liquid crystal. And then we learned that it's not just polymers. If you actually look at something called small colony variants of Pseudomonas, which are like mucoid colonies, they make a lot of polymers and biofilms. You essentially see that these things are crystalline, they're birefringent, whereas rough colony variants that don't make robust biofilms are not. And so how does this work? Cutting to the chase, what we learned is that essentially this phage is highly charged with stanninating. And so maybe a little like a magnet that has, a polarity to it and a charge to it. These things have an electrical field that repels things that are charged similarly. So if you took your magnet and I took my magnet, we tried to push them together, they would push against each other. And we think that that's what happens with polymers and phages. If they're free to rotate in space, they repel each other. And over time, what happens is the most favorable state for this to be in, particularly if there are cations there that can bridge this and mask those proteins. And we think, CF sputum is in a way, unfortunately, perfect for this, that this will form a crystalline lattice. And so I showed this to you 2 years ago when I gave a CF thing, but this is adding a drop of this bacteriophage that Pseudomonas makes to a solution of polymers. And this is what it looks like when it forms a liquid crystal.
Whoa. Wow. Was this an electron microscope or
a regular microscope with a polarizing lens? And
so I could my own sputum at home.
You could. And if you want to give it to us, we can see if you have PF phage.
Okay.
Yeah. Anyway, but the basic argument is that this is happening with CF sputum in individuals who are, and I'll show you data on this in a moment, who are infected with Pseudomonas strains that make this phage. That the reason why, or one of the reasons why sputum in cystic fibrosis is so tenacious is because many individuals have this phage. And that I would argue it's fundamentally an argument about entropy. And so Pseudomonas biofilms are tenacious for the same reason that diamonds are forever. It's this incredibly favorable entropic state that is really difficult to disrupt. Okay, so why should I care about this? Pictures are pretty, but how does this impact me? And so to figure this out, what we did is we took a strain of Pseudomonas called PA1, and we deleted this stage from this, and it's called PA1 delta PA4. And we grew it on a little piece of filter paper, 2 pieces of filter paper. That's what this Swiss cheese looking thing is. And, hopefully you can tell from there that the PA1 makes a robust biofilm with a lot of matrix. Whereas if we delete this stage, it does not. The biofilms are very wimpy. And then I don't have CF, but if I take my sputum, which still has polymers in it, it has mucins in it, it has hyaluronin in it, and I add a drop of this phage to that sputum, you essentially change that sputum from something with the viscosity of sputum into something with the viscosity of tomato paste. Like it essentially organizes it into a crystal, and crystals don't flow. Ice doesn't flow. You can actually do the same thing with cilia. So cilia line my trachea, and they essentially wave and they clear debris. And so in a healthy individual or a young person with CF, your cilia will essentially push up debris and bacteria and clear it from the lung. That's how this is supposed to work. But if you take those cilia and you grow them in culture in a dish, this is work done by Carlos Mila and Suju at Stanford, and you add our phages to that, essentially, just like with other polymers, because cilia are essentially long polymers, this phage will map them down and keep them from working. And so let me show you some videos of what that looks like. So Carlos generated cultures where he took cells from people with and without CF, and he grew them so that they made cilia and mucus. And as a way of measuring the ability of mucociliary clearance for cilia to move stuff, he took little beads that we've outlined here in color and added them and then tracked the movement of those beads over time. And so this is what it looks like if you take airway cells with cilia and mucus from somebody who does not have Pseudomonas or this phage. And what you can see is over 10 seconds, the beads migrate. This is what cilia should do. Healthy cilia should grab little beads or debris or bacteria and sweep them along. All right, everybody sweep with me. Yeah, there you go. Okay, so we're cilia and we're sweeping this stuff out the door, right? So what happens if you add phage to this? Well, not so much sweeping. Let's sweep here, right? Like this is the thing. And so we think that basically this phage organizes mucus in ways that keep cilia from being able to clear bacteria and other particulate matter like the beads we just looked at. Then the question is how common is this? And so for the first paper on this that we published at this point, 9 years ago, we took a group of patients with cystic fibrosis, and we took individuals who were infected with Pseudomonas PA, Pseudomonas aeruginosa, and people who were not infected with Pseudomonas, and we counted the phages that were in their sputum. Now, what we found basically was per 1 ml of sputum, which is about the same volume in the tip of my finger, we found that there were over 10 to the 7 copies of the phage. That means 1 with 7 zeros at the end of it. So think about how many phages that is, individual phages, that is in just that little bit of sputum. Essentially, the sputum in these individuals was jam-packed with copies of this phage. And then we looked at the sputum itself to ask, was it crystalline? And so we developed a special microscope that allowed us to visualize birefringence. And then we quantified that using a factor that the physicists had come up with called sine delta, and applied a heat map. So you can kind of see where birefringence is in the sputum sample. And essentially, there are long strands of sputum that are highly birefringent in this individual who's infected with Pseudomonas phage, PF4. But if we took a sample from someone who did not have PF4, their sputum was not birefringent. And this is just quantifying it. So people with Pseudomonas infected with this phage have birefringent crystalline sputum. Folks with CF who don't have Pseudomonas or this phage have minimally birefringent sputum, but if we spike in this phage, we can make their sputum birefringent again. So this sputum is necessary and sufficient to turn, this phage is necessary and sufficient to turn sputum from these individuals into a liquid crystal. So then more recently, we got explant organs from folks with CF who underwent lung transplant. And we looked at their tissues from their explanted lungs and asked, are they crystalline too? And essentially, this is the regular microscopy of those lung sections. This is the one of somebody who had Pseudomonas but not PF. This is somebody who had Pseudomonas but low amounts of PF. And this is somebody who had Pseudomonas but a whole ton of PF phage. And what you can see on this side, the heat map tells you how much birefringence is there. And so this person who didn't have PF, their lung section is not birefringent. On the other hand, the amount of PF phage you have determines how birefringent your lung is. And these are just data for a number of people. And so we published this just this year. Okay, so stepping back, what I've shown you so far, I hope, is that PF phage can partner, that bacteria partners with this phage to build crystalline biofilms. And that these things are happening, this is happening in the lungs and tissue sections of people with CF all the time. So why does this matter? And so to ask this, we asked whether or not the presence of these phages in bacterial colonies is associated with antibiotic resistance. So tobramycin, of course, is an antibiotic, as most of you know. And then CFUs are colony-forming units. This is how many bacteria are there per biofilm. And we increase the amount of tobramycin, and of course, there are fewer bacteria when you do that. But what we found is that if you take a small colony variant, a version of Pseudomonas that makes a robust biofilm, it's relatively protected from tobramycin. It grows a lot relative to a rough colony variant that doesn't make robust biofilms. That difference right there is its sensitivity to the antibiotics. If you take a version of Pseudomonas where we deleted this phage, or a protein from this phage, it behaves like a rough colony variant. If you spike in PF phage, now it behaves like a small colony variant, and it's much more resistant to antibiotics. So the question is how does this phage shield Pseudomonas from antibiotics? And so what some collaborators of ours at Oxford found was that essentially the red here is Pseudomonas. And what it does is it basically builds itself a shell, house of bacteriophages around itself. Maybe a little like an M&M where you have chocolate on the inside, the chocolate being the bacteria in this metaphor. And the candy shell is, it's terrible candy, but it's these phages that form like a crystalline shell around the bacteria inside and protect it from the environment. And this is what that looks like. Here's the Pseudomonas, and here's this crystalline network of bacteriophages wrapping around that bacterium. Then what we did is we looked at fluorescent antibiotics and asked, can they penetrate this? And so we took tobramycin again, and we labeled it with a fluorescent dye. And we found that it sort of floats around if you just have DNA or this phage alone. And you can't see it, but there's a bunch of bacteria there. If you add in DNA plus this phage, you make these shell-like structures, and essentially the fluorescent antibiotic can't penetrate them. It's stuck in the lattice-like network and can't diffuse across to the bacteria within. And so you need to be able to count this to do anything about it. So we took our phages and polymers and set up an MSTEP where we took a filter membrane, put these on one side, put antibiotics on this side, let the antibiotics go through, and then took an entirely different bacteria because we didn't want the phages affecting Pseudomonas. So we used E. coli. And what we found was that when there's a phage around, several antibiotics have a much more difficult time getting across a network of, again, this is sort of like a synthetic biofilm of phages and polymers. And so specifically, as trianan, amikacin, and meropenem had difficulty getting across, and tobramycin to some extent, and then ciprofloxacin was just fine. And this is notable because ciprofloxacin is a tiny neutrally charged antibiotic, where these other ones here are larger and have more electrostatic charge to them. And so to quantify this and to figure out what's going on, Kevin Chen, who was a postdoc in our lab that was supported by the CFRI, he took sputum from individuals with CF that Carlos Mila got for us from the CF clinic at Stanford. And he took antibiotics with this fluorescent tag, and then he mixed them. And he then used a laser to bleach a little square where there was no more fluorescent tag anymore. And he could count how quickly the fluorescent antibiotics diffuse back into that little bleached square. And that is something you can use to calculate a diffusion gradient. And so it looks like this over time. And so PFH basically influences how quickly fluorescent antibiotics can diffuse into CF sputum. And we found a difference with tobramycin, but actually not a difference with ciprofloxacin. If anything, the PF positive sputum samples, each of these dots is a sputum sample from a different person with CF. Essentially tobramycin gets blocked by this phage, whereas ciprofloxacin does not. I won't go through all the physics of this and the math, but what we basically found, we calculated something called calorimetry. So we calculated how strong the bonds are between phages and antibiotics. And what we basically worked out is that these phages form what's called a tectoid. The physicists already had described this for other things, a shell around the Pseudomonas inside. And instead of having chaos where antibiotics, polymers, and bacteria swim around, this creates structures that essentially block electrostatically charged antibiotics from being able to get across and kill bacteria. So then the question is, if you have Pseudomonas or care about somebody who does this matter clinically? And so to look at this, Liz Bergener, who is also supported by the CFRI, she now leads the Children's Hospital of LA Cystic Fibrosis Clinic. So she's a professor down there doing really great things with CF. And she's in Carlos Mila's lab and my lab. And she basically set up a study where she would collect samples, extract DNA from them, and figure out how much Pseudomonas and how much phage was there, and then look to see if that's correlated with all the numbers that we care about. FEV1, body mass index, exacerbation rates, that kind of thing. So what did Liz find? Well, in a pair of studies, one published in 2019 and one that just came out this year, Liz looked at 3 different cohorts. So in a cohort here at Stanford University, we found that about 2/3 of individuals who were infected with cystic fibrosis and were infected with Pseudomonas, this is 162 patients, 2/3 of them were infected with this bacteriophage. In a larger cohort of 474 patients from Denmark with CF, about, here again, 2/3, 69% were infected with this phage. And in a smaller Italian cohort of 26 people, here again, about 2/3 were infected with Pseudomonas. And what we find is that this is correlated with age. So if you break down each of these dots as a person with CF from the Stanford cohort from here at our hospital, Individuals with Pseudomonas tend to be younger. We all know that. In CF, you tend to acquire Pseudomonas as you get older and as your microbiome shifts in your lung. But if you look, individuals with low amounts of PF tend to be younger than those with positive PF or high amounts of PF who tend to be older. So, as we get older, along with everything else that happens to our bodies, if you have CF, you're more likely to be exposed to Pseudomonas. And over time, you're more likely to be exposed to strains that have PF. And the reason why this matters is if you then take all of these individuals and ask who is negative for Pseudomonas, who is intermittently infected and who is chronically infected, if you are infected with Pseudomonas, or if you have Pseudomonas showing up in your sputum and all with CF, and you have this stage, you're much more likely to have chronic disease than intermittent disease or negative status. So we think that this phage is influencing who develops chronic Pseudomonas infections. We also found in the study we published now 6 years ago, that essentially if you look at these individuals and you look at their bacterial cultures, they're resistant to Aspirin, amikacin, meropenem, and to a lower extent, it doesn't reach statistical significance for the other antibiotics like tobramycin. But the point is, these are the same antibiotics that were trapped in that filter assay that we set up. And so putting all this together, and I should add that this is the clinic patients, so I forget how many this is, but it's like 120, I believe. And each of these dots is different individuals. So the PF positive people are here in blue, the PF negative ones are here in red. And if you look at the slope of decline, if you're infected with strains of Pseudomonas that make PF phage and you have CF, your FEV1 over time, this is now a few years ago, so it's before HEMP therapy, but your decline in your FEV1 is more acute than if you're only infected with Pseudomonas that doesn't have this stage. And then if you break that down into people who have low amounts of PF and people who have high amounts of PF, we found again that the FEV1 declines in proportion to how much PF phage you have. Okay, so pulling this all together, what do we think is going on? So we think that you have somebody with cystic fibrosis. They make thick mucus that is hyperpolymeric, has a lot of polymers because it's dehydrated in a way. And so that concentrates the polymers. You also have a bunch of cations. It tends to be a different ionic milieu because, of course, it's a chloride channel, but it also affects things like bicarbonate. And what all of this does is it concentrates the polymers and it creates an environment where you have ions that both the concentrated environment with a lot of polymers and the ions facilitate the formation of these liquid crystals. And we think that if you don't have these, antibiotics can penetrate and kill bacteria. But if you have crystalline sputum, it keeps the antibiotics out, and the bacteria within can proliferate and mutate and grow. So this is something called antibiotic tolerance. And we think that over time, it selects for antibiotic resistance. And so you take people who are resistant to these antibiotics, and then, of course, you don't have good answers for their Pseudomonas infections. Okay, and then the other question we asked recently is how do people with CF acquire this? Here are 3 cohorts, again, from Stanford, Denmark, and Italy. And what we recently reported, again, this is with Liz and Carlos, in these 3 cohorts, you can see sort of a family tree of the bacteria that they produce. And so these are patients who are infected. Each of these endpoints is a person. So 162 people here at Stanford. And these folks are all infected with similar bacteria. These folks have similar bacteria. And if you look at where the phages are with this, the different colors of different versions of PF phage, essentially people who are infected with similar bacteria are also infected with similar phages. And what this is telling us is you don't acquire this phage from out there in the wilderness. The phage has been domesticated by the Pseudomonas. And when you get infected with Pseudomonas, you also get infected with the phage that it's producing. Does that make sense? So we think that essentially the phage has been domesticated by Pseudomonas and it uses it as a weapon, maybe a little like cat's claws or something like this. It's part of its armamentarium. And this is essentially evolved with the Pseudomonas that produces it. So we don't think you can acquire phages from other people. That might happen, but we think it's fairly rare. we think what's more likely is people with CF get infected with different strains of Pseudomonas, and if they get a strain of Pseudomonas that makes the stage, it's a particularly bad actor.
Sorry, question. The little chart on the bottom with the spirally branchy guy. What do you call that?
Yeah, let me explain this a little better. So if you take nowadays we can sequence a bacterial genome for about $150. It's cheap. It used to be very expensive. And so if we take each of these isolates from a different part of the world, so this is the group of patients that we're seeing in California here at Stanford, and there were 162 in this data set. If you take the genomes and you ask, just like if you were to take the genomes in this room, is anybody else Hungarian? Hungarian.
Oh, what?
You're Hungarian, okay. So if you were to take our genomes, you would find maybe a tree like this. You and I would be snuggling together. Our genomes would be closely related genetically. Whereas somebody from France or somebody from Russia, or whose family immigrated from those places, would be a little more distantly related, right? And that's essentially what we've done here with these bacteria. We've created a family tree for all of the different bacteria that we were looking at up here. And so of these, 65% of these bacteria have this PFage in their genome. And so then you can do a neat trick where there are different versions of this PFage, different family types, if you will. And I've assigned each of these, or Julie, who's the graduate student working on this, assigned each of these a different color. And what she found, like you have a whole area of blue, here you have a whole area of green, here you have a whole area of red. And so essentially strains of bacteria that are closely related to each other tend to have phages, PFages that are closely related to each other. And so if you were getting the phages from out there in the wilderness and they were just floating around infecting Pseudomonas, you'd expect this color wheel around here to be just a random assortment of dots. But the fact that it looks like this, where you've got an area of bacteria that are closely related to each other, and the phages are closely related to each other, means that the bacteria are evolving together with the phages. Does that make sense?
So that's like as the bacteria is growing and splitting, it's evolving slightly, and the virus is replicating during the same process than the virus.
Correct. And so basically this phage is a domesticated house pet of the Pseudomonas that produces it, but it's also a weapon.
Okay. Do you think the bacteria can see names like Fluffy to the phage or something like that? Do you think the bacteria, if you're calling it a pet, gives it like cute names like Fluffy phage or Midnight phage or Claw phage or something?
I bet it does. I don't know if you're a South Park fan and you've been watching the episodes recently with Satan in bed and
Phage.
Yeah, I bet there's some hanky-panky going on there.
Oh wow. Okay.
Yeah.
And then is it called a phylogenetic tree?
That is what it's called.
Oh, okay.
Very good.
Well, it looks really cool. I think it could have gone great on a press too. But we're making like a family craft, and I was like, that looks really cool. So.
Well, and that's exactly what this is. So, swap out the bacteria here for you and your relatives, right? And you would have a lineage you'd have your family tree, and you could map out things like hair color or the CFTR mutations onto a tree like this.
Yeah.
And there's a lot of interest in that kind of work, both in terms of people, but also bacteria.
Okay, cool. Thanks so much.
Yeah. Okay, I'm going to skip over that. This is just to say we did a separate study in wound patients. So we followed 113 wound patients for 2 years. This was another clinical fellow in our group. And here, a fraction of them, 39, were infected with Pseudomonas. And again, about 2/3 had this stage. And what we found is that chronic wounds, if you didn't have this stage, you cleared your wound infection in half a year. But if you did, it took over 2 years to clear those wounds. And so stepping back, how do I understand this? I'm a biologist, and I think about, so the CDC has all these websites about parasites, right? And oftentimes parasites have complicated life cycles. So this is a snail that's infected with this little parasite. The snail can infect little crayfish, and then it can also infect people, and it's part of one big life cycle. And so the way I think about this as biologists is that this phage is a parasite of the bacteria, but it's also a parasite of us. And so if you're infected with Pseudomonas, you're infected not only with Pseudomonas, but also independently in a way with this virus that partners with Pseudomonas to cause disease. And I think thinking about it in these terms, where you have Pseudomonas and you have this phage, is potentially helpful if having strains of this phage or strains of Pseudomonas that make this phage are such bad actors. So then the question is what are we going to do about this? And so what we realized is that this phage tends to have a conserved coat protein, which is its shell. And so one of our immunologists in our lab, Christian DeVries, noticed that there was a lot of conservation between this coat protein and asked, could we make a vaccine with this? And so we developed it's very difficult. Now there are better airway models for cystic fibrosis. But one of the reasons we've worked in wounds is because historically there have not been good models. You had to go into pigs or ferrets. We did this work in mouse wounds, and we infected them with Pseudomonas. And if we re-vaccinated them with this phage, we could actually prevent Pseudomonas infections. And then if we took monoclonal antibodies against this phage, then dosed the mice with those before challenging them with bacterial infection, we could actually prevent the Pseudomonas infections. And so what we think is actually going on here is the reason why there's no vaccine against Pseudomonas or, non-tuberculous mycobacteria or many other bacteria is that they have a layer of slimy polysaccharides around their surface. And that keeps things like macrophages, which are cells in your immune system that gobble up bacteria, from being able to stick to them and eat them. What we think is going on, I showed you earlier that shell that bacteria make of phages around them. Phages are protein. And so it's a little like having a rock climber. Your antibodies can climb a rock wall, can climb a protein wall because they can stick to it. Unlike, say, a mossy, slimy, seaweed-covered wall, which is your polysaccharides, where it's just too slippery, you can't bind to that. And so what we think our antibodies and our vaccines are doing is sticking to phages that are on the surface of the bacteria and allowing them to then be engulfed by macrophages. The other thing that we find is that if we pre-treat cultures with our antibodies, that antibiotics can get through better. We've also been developing what's called little peptides, so antimicrobial peptides that stick to this phage and that we're combining with antibiotics to let them get through. And that is what one of our CFRI-funded fellows is doing. And so basically in these two ways, with both vaccines, but then also small peptides that we think we can dose with antibiotics, our goal is to shatter the crystal, if you will, to make antibiotics more effective. Well, first of all, maybe to prevent Pseudomonas infections in CF, but then also, to bundle these with antibiotics to make them more effective at penetrating CF sputum. And that's the vision. And so I would offer this is a kind of phage therapy, if you will. And so the conclusions from the first part of my talk are first that Pseudomonas partners with these phages to build biofilms. This phage can promote chronic lung infections in people with CF. And then this phage is a novel biomarker and potential therapeutic target for things like small peptides and vaccines. And so we're currently evaluating that vaccine in pigs, and I'm hoping to have more data on that soon. Okay, before I go into the second talk about, I think, lytic phage therapy and maybe what some of you thought you were going to immediately hear about, any questions about what I've talked about so far?
I have a lot of questions, but have other folks have questions.
I can also come back at the end.
I have a quick question.
Yes.
Do funguses have phages as well, or just bacteria?
Really good question. Fungi do have bacteriophages. They are called mycophages. Yeah, but we don't know. I haven't seen anything that implicates those, for example, in Aspergillus or other types of fungal infections. But basically, bacteria use bacteriophages to do everything from transmit antibiotic resistance, transmit toxins, and I'd be surprised if fungi don't do some interesting things with them too. Okay, so let me dive in and tell you the second story I was going to tell you, and this is about lytic phages and how they can protect people with CF from bacterial infections. And so I mentioned there are two types of phages. The lysogenic guys are the ones that we've been talking about. Lytic phages straight up just kill bacteria, and then they move on to their next prey. And this is what that looks like. This is an image of a bacterium. I think this is E. coli. Yeah, right. That's been with phages, and I could watch this all day. Let's see that again. It's just sitting there unsuspecting, and the phages build up inside of it and lyse the bacteria. And so I think we and many others are excited about using phages like these to clear bacterial infections in people with CF. And so what's been happening over the past, I would even say century of phage therapy, phages were discovered just over 100 years ago, is that people have been trying to develop excellent phages that they can use to treat bacteria. And one of the ways that has become more practical over the past, say, 10 to 15 years is what's called personalized phage therapy. And what you can hear is you take somebody's bacterial isolate and you find, we have a library of about 30 phages, Pseudomonas phages in our lab. Other groups have as many as a few hundred. But what you do is you identify phages that work against that particular isolate that works for that person, and you use that to treat them. There are also hybrid models where you have a group of phages that are known to work well together, and you essentially test people and identify cocktails that work well. And this kind of work is being done by governments in other countries. So Phage Australia, in England, in the United Kingdom, in the Georgian Republic, which used to be part of the Soviet Union, they do this, and in Belgium. Here in the US, we don't have that. It would be a wonderful thing to advocate for. Instead, we've got a bunch of academic groups that have been trying to develop phage therapy. The problem is it's very expensive and difficult to get up and running. It's essentially like running a small pharmaceutical company out of your lab. And so this has been challenging to do without a whole lot of government support. Nonetheless, there are good indications that this can work. So this is a study out of Belgium that came out, I guess, maybe a little less than a year ago, where they looked at 100 consecutive cases that were treated with phages that were developed in Belgium. And what they found was they were able to eradicate the bacterium in just over 60%. And they saw clinical improvement in about 70. And they did this together with antibiotics. This isn't cystic fibrosis cases. What this mostly are people who had one bacterium or maybe two bacterium, and they had like a joint infection or some other kind of thing where they could go after just a small number, ideally one bacterial pathogen. This approach has been working in cystic fibrosis. So this is a really super cool case from a couple of years ago where this was treatment for non-tuberculous mycobacteria. So an organism called M. abscessus that was done in Pittsburgh. And what this group did here is they essentially engineered the phage so that it couldn't even infect other bacteria. They essentially got out any bad genes from that bacterium. And using this engineered bacteriophage, they were able to clear non-tuberculous mycobacteria from a 15-year-old girl with cystic fibrosis who had already received a lung transplant. And she did well with this. She cleared her non-tuberculous mycobacteria. And to my understanding, that same group in Pittsburgh and other groups that are aligned with them have now treated, I would even say I think it was over 20. They had a case series that came out recently. And I think now they're probably higher than that. I would guess 30 or more. And again, this hasn't worked for everybody, but they've had really encouraging success rates, which is exciting. The thing about it is, though, all the cases that I've been telling you about are typically with a single bacterium. And the problem with something like cystic fibrosis, or indeed most of the chronic infections that we would want to treat, is it's not like there's one bacterium there. So you essentially have, in the context of a CF lung, many different bacteria. And what that means is because phages are so specific for the bacteria that produce them, you would need a mirror image of all of those kinds of phages, or a phage that at least has a broad host range and can infect all of those different bacteria. And then this gets especially complicated when you consider that each different patient with CF has a different group of bacteria. And most of the time from CF, I mean, people get infections from other people. And this is why they don't do things like CF camps anymore, or why we don't necessarily want to shake hands. Bumping elbows is better.
But we do retreats.
You say it again?
We do retreats.
But we do retreats. And, but the thing about it is that, in order to essentially treat all of those different individuals who all have different panels of bacterium, it's very difficult to come up with a standardized cocktail that can work for everyone. And so I was particularly excited to see this paper come out earlier on this year. This is a group out of Yale led by Jonathan Koff, who is a pulmonologist. We call himself a CF physician. And here they treated 8 people, if I remember correctly, with different cocktails of phages. So this is personalized therapy. They essentially took clinical isolates from each of these individuals, paired them up with phages from their library, and then looked to see what they got. And what they saw, so CFUs is colony-forming units. Each of these is a person that was treated with personalized phage therapy. And they were able to knock down the amount of Pseudomonas in these individuals by, in some cases, several logs, in some cases, less than that. They didn't eradicate Pseudomonas in any of these patients, but this is really encouraging. And what's also encouraging about it is in many of these individuals, along with treating their Pseudomonas, they observed an improvement in their FEV1. Now, after you stop treating with the phages, it seems like the Pseudomonas bounces back in many of these cases. But I would offer that this is really encouraging because it's what progress looks like. There's a company called Biomex that has a phage therapy clinical trial going on right now in cystic fibrosis. They have a cocktail of engineered phages, and what they do is they identify people to treat with this, and they did this, I think, for 2 weeks. This paper just came out maybe a month or 2 ago. And here again, this is log CFU, so it's a 10-fold change. And the folks in blue are treated with phage. The folks in orange got placebo, so a preparation that didn't contain any phage, and that's to control for the placebo effect, right? This idea that you're getting treatment and you are going to do better. And so there's perhaps an improvement here. There were too few patients here. This study was really about safety and tolerability. And they're not eradicating Pseudomonas, but the fact is they're getting traction. And to use my rock climber metaphor, it's a little like watching somebody climb a wall. It's a slippery wall. It's covered with moss and algae and whatever, but we need to climb it. And watching somebody, get up, 20 feet, 30 feet, even if the wall is much bigger, I think is exciting because it is progress. So to step back for a moment, I would offer that phages have a number of advantages. They're strain and species specific, they're effective against antimicrobial resistant bugs and biofilm, and they're extremely well tolerated. People don't get sick from phages because they don't tend to infect eukaryotic cells and replicate and cause disease. However, phages have a bunch of disadvantages that we need to address. One is this narrow host range, because typically phages only work for a subset of Pseudomonas. Bacteria can develop resistance to phages in the same way that they can develop resistance to conventional antibiotics. And then our experience in the phage therapy field tells us that we need to combine phages with conventional antibiotics to get the best bang for our buck, but we don't really know how to do that. Phages can have antagonistic or synergistic. They can basically, like people, they can either work well together or they can fight. And what you want is a team of phages and antibiotics that are going to come and fight for you. And so the question is how to identify those. And so one question we've been asking in our lab is how can we do this better? How can we make broadly effective, off-the-shelf phage therapy? And I won't go through all these different studies, but essentially we're not the only ones working on this. There are groups that are engineering phages to get them better. There are groups that are using artificial intelligence to identify phage genomes that are more likely to be successful. And then there are a lot of groups that are trying this empirically. And so what we've been asking in our lab is, here again in the second half of my talk, and I'll make this really quick because I think I'm almost out of time, are 3 questions. What determines phage host range? How can we prevent the emergence of phage resistance? And how can we identify phage and antibiotic combinations that work well together against cystic fibrosis clinical isolates? I'm going to skip this part of my talk because it's fairly molecular, but basically we did a bunch of studies to identify what determines whether a particular phage can bind to a particular bacterium. And I'm going to cut to the chase and tell you that it's the receptors on the cell surface. This is a little bacterium, it's Pseudomonas, and it's big and slimy, but it has a couple of features that phages like to stick to and use to get in. It has a little flagella that it uses to swim. It has something called a type 4 pilus, that's a grappling hook that it uses to attach to your kidneys. And then it has a wall that's made out of something called lipopolysaccharide. And then it has these other oligosaccharide antigens. And as far as we can tell, for Pseudomonas, all of the phages that we've ever identified, which is a lot, all use one of these 4 structures. And that's what we figured out. So then the question is how can we use that information? There are essentially 4 different receptors on Pseudomonas to make cocktails with broad range. Can we combine different phages that target these different receptors to essentially cover all the bacteria with it? And so Kevin Kim was an infectious disease fellow in my lab, and he had this idea that, I don't know if you guys know much about HIV, but nowadays, if you were to get infected with HIV, say, by a needle stick or in the hospital or what have you, they would give you a cocktail of drugs that each works in a different way. And using that cocktail, we can essentially get most people undetectable. It turns out that if you have what's called selection pressure, evolution happens, but if there are too many things fighting you at the same time, the bacterium, or fighting the bacterium at the same time, or for that matter, the HIV virus, it can't evolve around it. And so Kevin thought we could use the same strategy against bacteria. And I'm going to zoom through some of these technical bits. But what he essentially found was that this was the case. If you take different phages, which is each one of these things, and you look to see how they work together, you can identify what we've been calling complementarity groups. And so essentially, if you pair phages that target the type 4 pilus with phages that target lipopolysaccharide and phages that target flagella and OSA, you get quite good combinations of phage therapy. And to show you what this looks like, we measure bacterial growth by looking at turbidity, something like OD600. And if you just take bacteria and you grow it in a dish, you see this red line here. If you add 1 phage or 2 phages that use the same receptors, you might delay that growth a little bit, but not by much. If you take 2 phages that use different receptors, you can slow that growth down to like 30 hours. But if you take 3 different phages that use 3 different receptors, you can essentially prevent the emergence of resistance in that bacterium over 30 hours, which we think is really exciting. And for some reason, also this combination of 3 or 4 drugs is essentially the number that people use for HIV cocktails. And so we think there's something really cool going on there about how evolutionary pressure works and all of this. And so then the question is can we do this with cystic fibrosis benefits? And so we got clinical isolates from, again, patients at the CF clinic at Stanford, and we treated them with individual phages. So orange means that the bacterium was killed, and black or gray means that it was not killed well. And so individually, some of these phages worked well against these different clinical isolates, but a cocktail tended to work really well against most of them. And we could actually do this against individual bacteria. We could also combine bacteria and treat them all at once. And the cocktail, with what we've been calling our suppression ratio, worked the best. And Kevin developed 3 cocktails that each, over around 80% of the cystic fibrosis clinical isolates. So we have 174 Pseudomonas isolates in our repository. And we find that we can make cocktails that kill the majority of them, which is remarkable because most of these are resistant to many antibiotics. Actually, this is a drawing. This is what happens if you bring your 15-year-old daughter to a CFRI meeting and talk about phage therapy cocktails. This is what she draws and gives you afterwards. I thought that was really pretty great. Then the question is, can we do better? Can we combine phages with antibiotics using this same method? And so essentially people have developed these assays here, which look at the interactions with the antibiotic on one axis, the phage on another axis, and you look at how they collaborate. What we did is develop an assay where we can turn that number into just a single digit and then looked at a heat map. And so if we take our different phages from the previous thing that I showed you and all the different antibiotics that work against Pseudomonas, what we find is that individual phages tend to have different relationships, but patterns emerge. And we think that antibiotics and phages tend to target the same structures on bacterium. It's like a knight who's wearing armor. There are only so many points where the armor comes together and creates sort of gaps in defenses, right? If you're going to have a flagellum, that's a point of weakness. Or if you're going to have lipopolysaccharide, that's a point of weakness that either phages or antibiotics like tobramycin can target. And so what we realized is that some antibiotics work better with all of the phages, and some are hazard and don't work well at all. In general, we find that beta-lactam antibiotics, things like meropenem, aztreonam, that these things work much better with most of our phages than other antibiotics do. And then there are some antibiotics that are really just terrible in terms of interacting with phages, and we can talk more about those later. Anyway, then what Kevin did is took all of the 174 clinical isolates in our data set, or I guess this is 153 of them, and then compared our cocktail, looked at aztreonam by itself. As you can see, almost all of these isolates are resistant to aztreonam. These are all patients, from our CF clinic who've been getting antibiotics for a long time. But if you mix them, the combination of our cocktail plus aztreonam actually works really well for most people, including against biofilms. And so if you combine our cocktails, we were able to get up to 96% killing of our clinical isolates with the combination of phages and antibiotics. And we essentially now have 3 cocktails that we can treat, that we can use, that at least in vitro give us, over 95% killing in terms of different bacterial isolates. So we're really excited about this because we think that this may be a path forward to how to make off-the-shelf phage therapy so that we don't have personalized phage therapy for a single isolate. And so we can do better than just taking down the CFUs by a log or two and maybe begin to optimistically talk about clearance. And so the hope is that we can essentially bundle cocktails of phages and antibiotics and develop this as treatments for Pseudomonas and potentially for Staph aureus and other bugs. So to wrap up this part of the talk, I would offer that, I didn't show you this, I skipped through it in the interest of time, but receptors on the surface of bacteria determine what the host range for a particular phage is. That seems pretty intuitive. Combining phages from what we're calling complementarity groups, maybe a little like an HIV cocktail, creates effective cocktails. And hopefully that's fairly intuitive also. And then I would offer that phages are a frontier in the treatment of CF lung infections. And I'm incredibly excited about the trial going on by Biomics. The CFF is also supporting a trial. And, there's a lot of different investigators around the country and around the world that are working to develop phages for use in CF. And I just think it's a neat time to be doing this work.